This is part 2 in our series on transcription and translation, and here we are focusing on post-transcriptional modification, RNA splicing. In order for a pre-mRNA transcript to be successfully converted into mature mRNA and then translated, processing must occur. So in our previous video, we've covered that a GTP cap will be added to the 5' end of the pre-mRNA, and by the time it gets transcribed, there will also be a 3' poly A tail that's added to prevent against exonuclease degradation. However, the pre-mRNA transcript at this point still has introns and exons included. So splicing is the process by which introns are removed, leaving just exons. So splicing helps us explain why the human genome has about 20,000 coding genes, but up to 75,000 functional proteins. Alternative splicing results in different transcriptional byproducts. So we will just go ahead and jump right in here. Proteins that facilitate splicing are called small nuclear riboproteins, and they're often abbreviated SNRNPs. These proteins recognize both the 5' and the 3' splice sites based on certain nucleotides present. So the GU here at the beginning of an intron and an AG at the end of an intron both are indispensable and invariant for this process to occur. The first step is small nuclear riboprotein U1 binds the 5' splice site. Then branch point binding protein SF1 and U2AF proteins bind the branch point and the polypyrimidine tract near the end of the intron. U2, another protein, then displaces SF1 and U2AF at the branch point, and this forms the bulged A. The spliceosome is a tri-SNRNP complex, which includes U4/U6 as well as U5. This is recruited and releases U1. In a trans-esterification reaction, the 2' hydroxyl of the branch point attacks the 5' splice site at the phosphodiester bond. This cleaves the intron-exon junction here and forms a looped structure called the lariat. Then another trans-esterification reaction occurs where the 3' free hydroxyl on the first exon attacks the 3' splice site at the phosphodiester bond. The intron, now a lariat, is released and the exons are ligated together. A protein called the exon junction complex will sit on the ligated site as a physical marker that, hey, this is the location of a mature exon-exon junction. So here's just another way to view this reaction taking place. You have the hydroxyl group here at the branch point. In a trans-esterification reaction, it will attack the 5' splice site phosphodiester bond. This creates the lariat or looped structure and frees up this hydroxyl group here on the end of exon 1. The hydroxyl then attacks the phosphodiester bond here on exon 2, completely freeing up the lariat or the intron, and the two exons are then ligated together. Certain cells express different proteins that might help regulate various splicing events. Some proteins might promote intron exclusion, while some might promote intron inclusion. This is really complex and very context dependent. You can see here that one gene could make 3 different proteins based on alternative splicing events. So the first protein here has all 5 exons, while protein 2 is missing exon 3, and protein 3 is missing exon 4. Now the pre-mRNA transcript has been capped, it's been spliced, and a poly A tail has been added, and so now it's just ready to be translated. So our next video will cover just that.